Hi guys, so we are back to the lecture. All right, so this is the one that we left earlier. This is the 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 discipline to analyze the chromosome is called the cytogenetics. So basically, this one is a combination of two principles. One is cell, the other one is genetics. So basically, this one is to look into more like a molecular variation related to the chromosome variation. So that karyotype is an earlier. It's also that's part of it to look into, but karyotype is more like the resolution is different. The karyotype is looking to the uh, more like the counting of this chromosome, and we do be we are able to identify the short arm, long arm, and also the number of chromosome. But with cytogenetics, it's more like a broad analysis to see more this chromosome variation in different form. So that's that. So just figure kind of give you an idea that this is the principle. The scientist in the cytogenetics, it's called the cytogenetics, right? All right. So by looking into the karyotype, that we are able to identify a lot, right? So we can sort these chromosome. So for example, here is typical one that you can see uh, 23 pairs of chromosome that we are supposed to see. Uh, but something like this one, you can easily identify that something is not right here. We have 1 to 22, and this one is X, Y, and X. Sorry, X and Y, right? And then we see something here. We see extra 21. So this is a type of the chromosome abnormality that we can identify one extra. Uh, so there are different type of the chromosome abnormality, and we are going to talk about not all, but some, but selective those that's more like commonly seen in the clinical situation. For example, the uh, polyploid, this one is with the extra set of the chromosome. So with this one, the person will not only have 23 pair, it will be 33 times say 3, right? Uh, a lot more chromosome in the cell. We also have this aneuploid that is not just this extra set, it's only extra chromosome for a type, a chromosome. For example, a typical one is the trisomy. Trisomy not very commonly across the 23 chromosomes. We do have some trisomies or extra chromosomes in the sex chromosome that we mentioned earlier. Um, but in the autosomal chromosomes, uh, extra or extra chromosome can sometimes lead to the uh, sudden abortion. It will not; these cells will not survive. Only those selectively, maybe 21, trisomy 21, trisomy 18. 21 is the most common one. The 21, let's look at what's 21 here. So this is the typical one, trisomy 21. That the one, why is this one will survive even though this is still abnormal? It's because that during the development, these proteins doesn't really, the amount of protein provided by 21 is not a lot first. And the second is that this protein don't affect the development of the fetus that much. So even though you have extra protein, extra sets, it doesn't really affect the fetus development that much. And so the fetus will survive, get delivered, but only when the after the birth, this protein, even though not a lot, but plays an important role. One of these protein here is related to the several things. One is very common known as the amyloid beta. The amyloid beta, extra sets of this amyloid beta can lead to the more production of this amyloid beta peptide, and that peptide can increase the amount of accumulation in the brain, and that could lead to Alzheimer's disease. But only when the person is aging, then that effects will become a fetus, like fatal issue, right? Otherwise, the patient will basically survive. And this related is that this affects a lot of this neural development and affects the brain development. So, but this development usually happens after birth, not before the birth. So the patient will, with the 21 trisomy, it's not the chance to have this trisomy is basically supposed to be equal across different chromosomes. But the reason that we see more in 21 and a little bit more in 18 is because these proteins survive, these chromosome extra proteins survive after birth. So that's that. So here is a list of the chromosome abnormality that you can see that in the poison. All right, so we talk about this one, trisomy, and monosomy is the not extra but absent chromosome. So this one, for example, we will have typically people have 46 chromosomes. With this one, we have 47. Like this one here, we will have 47 chromosomes, or we may have like this one, we may 46 minus 1 is 45 chromosome. We may have a deletion, duplication. This is like the amount of the chromosome is not changed, but the one segment is either lost or get duplicated or get translocated. Translocated must not be like homologous, right? If it's homologous, it's exchange, it doesn't really make any difference. So this translocation only when this translocation occur between non-homologous pairs will make a difference. So for example, this one and the other one, that will make a difference. Otherwise, if this segment, that segment switch, well, if the switch is basically the same amount, well, you probably we will not see any difference unless the amount is different. So translocation, inversion, so you basically that have the translocation, but insertion is inversed. And sorry, isochromosome. So that's the chromosome with identical arms. Say P arm, Q arm are identical. And also ring chromosome, the chromosome delete the telomere, and then their two ends adhere together. So this is another abnormality of the chromosome. So that's several types here. So we will go through this and to talk about the for example trisomy, and we will talk about the trisomy 21, trisomy 21, and then we will talk about how clinically, how do we prevent this one, even because this is the most common type of the chromosome abnormality. We will then talk about the translocation, and we will talk about two type of translocation, and how is that can affect, because translocation, if these two translocated, the total amount of chromosome, the gene may stay the same, so it probably won't cause any issue. But if this translocation cause the unbalanced result, then this may cause any issue. So we will talk about that. So that's probably it. And so let's look into it. This is the polyploid. The polyploid is an extra set. So it will become 3n. N is 23. N is the number, right? It's the number. So typically cells has 2n, not n times 2. Times 2 means identical copy. This 2n, not identical copy, because one copy from the father, the other copy from the mother. So we have 23, 2n, 46. But these 2 and these are not identical. All right, so now here we have the 3n. That's extra sets. This, how can we have this one? So this happens because when the cells conduct this meiosis, they may not split well into two different daughter cells, right? So you may have, let's see if I have something like this one here, for example, like this one. So say this is the first split, second split, and here you have polyploid, right? So here you split. You're supposed to split two, but you got one empty, right? And these two got extra. Or you may have something like this one. The first split division is fine, the first meiosis. Then the second meiosis doesn't really split. This condition, this, when they don't separate well, they supposed to be disjoint, right? Like disjoint, supposed to be disjoint. This is the chromatin supposed to be disjoint, but they do not. This condition is called non-disjoint, uh, and that is the cause to cause this chromosome abnormality. In this case, if it only happens in one type of chromosome, this is called uh, trisomic, right? And this will become monosomic, trisomy and monosomy. So that's that. So that's the polyploid. 2/3 of all these triploid, like 3n, caused by the fertilization of one egg and two sperm. So that will cause the extra set, because it's supposed to be one egg and one sperm, but now you have two sperms. This would be S right here. The other could be the diploid gamete. So if this is the not just one chromosome but the whole set, it will be a diploid gamete. And sorry, not this one here. So polyploid almost all lethal, so we don't see this one get delivered. If it's got delivered, may survive only a few days. So we don't, even though this is a condition, this can happen, but usually we don't see it survive. But in single cell, it can be polyploid. So liver cells and can have these polyploid sets of this chromosome, which is, in these cells, they are probably be okay. They'll be okay. So this is extra set, right? You have the extra sets. So you have 3N, not just 2N in a regular person. All right, so this is, even though this is a condition, but this is rarely seen. These patients won't survive. Most of these patients won't survive after birth. Another more commonly seen, in the living like a human being is this aneuploid. So this one is the aneuploid include trisomy and monosomy. So it's either extra one or chromosome or missing one chromosome. And the cause of this one is the non-disjunction. So as I mentioned before, is the split is not well. And so this supposed to be is joint, should be disjoint, separated, but it's non-disjoint. So that's the cause of this aneuploid. And the common one of this extra set trisomy occurs in trisomy 21, trisomy 18. The most common one is 31. So this is from the most common, second most common, and the third most common. The others may happen, but their extra sets of protein will make this organism become not able to survive. But in 21 particular, organism can tolerate it, and so it will carry it after the birth, and even longer after the birth. So for the daily life function or maintenance function, these extra sets are okay, but typically they will lead to some defects, making the function become impaired, and the lifetime usually is shorter with trisomy. So that's that, and this is the example of the aneuploid, and the condition is caused by the non-disjoint chromatids. So for this different type of the chromosome abnormality, we generate the way to quickly describe it. So we typically that when there's a disease, right? When there's disease, these usually go with the person's name after the person who discovered this disease. But for the chromosome abnormality, these will make it too difficult to memorize all these names. Besides, these are more or less not very common condition, and the condition can be a little bit randomly happens, right? So they come up with this easy way to identify each condition, and so that's written here. Basically, that we will first put the number of the chromosome. 46 is the typical normal number, 2n. We may have 45. 45 means that it missed one. So this is the monoploid. Monosomy. Monosomy, that's right, monosomy. and 47 means actual trisomy, right? And we may, in addition to this, we will also put the sex chromosome following that. So if it's XY, it's healthy, it's male. If it's XX, it's healthy, it's female. If it's X, then it means one. And so by looking at this single X, you know that it means one. So it's the monosomy 45 chromosome only. And this is one that we mentioned very early, like the sex genetics that we mentioned about this one. This is a female, but it only has one X, not two X. So the development is not fully developed to the sex characteristics. So this one is also called the Turner syndrome. This is a relatively common like sex condition. We may also have XXY. We have extra Y or extra X, right? So it's that condition. And this is also not like uncommon. This is maybe second to the Turner syndrome, uh, Klinefelter syndrome. So we can have the name, right? Name of this disorder, but uh, it will be easier to just use this. It's called the shorthand, chromosome shorthand. This label to label exactly what is the condition. The syndrome usually is to describe that this phenotype. Syndrome is usually a phenotype that contains, the spectrum of the phenotype, but the genotype would be it would be better to identify to be explained to be presented by this uh, chromosome shorthand. So with this one is one extra, right? So you have 47, one extra XYY, 47. Now with this one, say 46 XY, with just this, you would think that this is normal. And so to tell, it's not exactly lost the entire chromosome or extra chromosome. Say, for example, like this one here. This one is 47, so you know it's extra. Now you have XX. So this extra is not related to the sex chromosome. This is related to what? Plus 21. So you know that it has 1021 extra. And so this is 21, trisomy 21 Down syndrome. And the female XY. So if the number is not different from the 46, say 46 XY, so everything. So now what is the disorder will be identified here? It would be delete is missing one, missing part of the 7q. Q is a long arm, so it will be the missing 7 chromosome long arm Q. So this is a deletion, loss of the part of this chromosome. We may have a translocation, say this one. So this one, this is 46XY. So from here, the number of the chromosome is not different. But here we have a translocation. T refer to the translocation. Translocate what? 7 and 9. So 7 and 9 chromosomes. Which part are translocated? P21.1. So that segment is translocated between 7 chromosome P21.1 with the 9 chromosome Q34.1. So that's that. And now we have not just one extra, but two extra. So it's not 46, it's not 47, it's 48. And these two extra are here, X, X, Y, Y. So if you have 40, if you see 48 XY, then you will expect to see some plus something here to match with this 40, the number 48, the number of the chromosome. So that's that. So this one is very easy. If we have the exam, probably I can have a quiz on this one here, right? So we can have for example, 47xx plus 21, and I can have a list of options that say trisomy 21, or say Turner syndrome, or maybe the deletion of this or missing of that, translocation of this, translocation of that, something like that, right? So that's that. All right, so here is a summary of this key concept from this segment. The chromosome abnormality can be viewed as the extra set, which is ploid. This usually not happening because the infant won't survive after, even before birth. The more commonly seen one is the aneuploid. The one of these aneuploid is the trisomy. Trisomy, the most common trisomy is trisomy 21, followed by trisomy 18 and 13. Aneuploid can also happen with this extra or missing chromosome in the sex chromosome. So we know there are 5 related sex condition. X0, that's missing one, Turner syndrome, triple X, XXY, XYY, XYY, so something like that, right? So this is part of this aneuploids. And this is one that we actually mentioned about very beginning of our lecture. And the cause of this aneuploid is due to the mitotic non-disjunction. So that's that. All right. So these all these condition, if it's sex condition, this usually it affects the sex characteristics, but the mental wise is not really affected very much compared to the trisomy. So trisomy 21 is the most common aneuploid. So let's look into this one. And it in a way, trisomy 21 is very easy to identify because we can see it. Not this one here. We can see it just by simple karyotype. We can see it here, 21 extra chromosome. So if we can detect this early, so this is not something identify a gene or identify a mutation of gene. This is not a gene. To detect one gene would be more modern technology to do that. This is to identify just one extra set, one extra chromosome. So this is easier to quantify, and this technology has been a while it's been a while. So people can simply do a karyotype and to identify if this, say, egg, right? If it's just the egg, and maybe it's the if this is the fertilized egg developed, and if it's only in a very early stage, and if we can know this early, maybe the parents can have some preparation for it. So that's that. So that is, and we know that also research over this long period of time, research has known that the frequency to have a baby with trisomy 21 is proportional, not proportional, it's depending on the maternal age. So you can see that if the mother's age is less than, say, 30, or say even 27, something like that, the risk is not exactly low, but it's not change. However, when the mother's maternal age is 30, 35, 40, 45, then the chance, the risk, the chance to have a trisomy in the baby become trisomy 21 is getting higher and higher. So that's that. And so it will be a good idea for the, we probably won't need to do it because we know we probably won't need to do a fetus chromosome test for all pregnant women, but for those at the risk in at the age which has a higher risk to give a trisomy 21 infant would be a good idea to do some fetus chromosome test. All right, so what are the technology that we can use for the fetus chromosome test? There are two major ones. So the first one is the amniocentesis. This amniocentesis basically use a needle and get the amniotic fluid. And because this fluid may contain some discarded cells from the baby, so we can use that to, and all these cells are divided from the fertilized egg. So it could be a good way to see what's going on with the whole body, right? The fertilized egg. And so that's a good way to do that. Another technology is called the uh, chorionic villus sampling, or short for CVS. And this one detects the cells from the chorionic villi. We talk about this chorionic villi when we talk about the pregnancy, and so that's also a good one. However, so there are two types. Which one is better? Uh, the uh, the There are pro and con for each. Amniocentesis is one that's uh, relatively safer, and but the condition is that the situation is that we will need to, because the cell number is less, so we need to culture these cells, and may take almost a week to grow enough number of the cells to conduct the karyotype. As for the chorionic villus sample CVS, the advantage is uh, it's faster, because here we can collect a larger amount of cells, and besides, we don't need to wait until there is enough of these cells. For the amniotic fluids, there is a period of time that's provide a good amount of the fluid space for us to get the sample. So there is, however, this one is less accurate because these cells may include mother cell. So but this one, the advantage is faster because we don't need to do the cell culture. All right, so let's look into the amniocentesis, this technology. So this one performed in about 18 to 20 weeks gestation. And by doing this one, we need to have an ultrasound, needle enter into the amniotic fluid, and the cells will be cultured for about 10 days, and to grow about 20 cells for karyotype. and then there is a risk. Nothing. This is not like totally safe. So the risk is a miscarriage, and so when patients consider or the doctor recommend a woman to take this, they need to weigh which risk is higher. Only when the risk to have a trisomy 21 is higher than the risk to cause miscarriage by the amniocentesis, then doctor will recommend. Otherwise, if this procedure provide a higher risk than the risks to develop the trisomy 21, then this is not recommended. So the earlier data, and a lot of the institutions still following this earlier data is because that it's safer, right? Even though the risk may not be so high with this technique, but just play the safe side. So the earlier data is that the risk to cause miscarriage by the amniocentesis is 1 in 350. So get the number first. Remember the number 1 in 350. So let's look into the trisomy 21. 1 in 350, right? 1 in 350. So this is risk to cause by this technique to cause the miscarriage. If and when which age and 1 in 350 is wrong here. So let's draw a line. Let's draw a line. Say somewhere here. How about that? Somewhere here. Somewhere here. Right? So that, all right. So that is the age. So that is the risk, 1 in 320. So if the patients is more than that, say this is 35 years old, say 37 years old, with this woman, she is going to have the baby, and her risk to have a trisomy is this one here, is say 1 in 400, for example. Say 1 in 400, right? So 1 in 50, 1 in 100. So say this is 1 in 400. So here's a chance is 1 in 400. Sorry, I was wrong. 1 in 350 is not there. 1 in 350 is supposed to be. Let's just move it up, not here. 1 in 350 not here because 500,000, 1 in 350 should be around here, all right? So it should be around here. And so this one would be somewhere here. And so if the woman say this is say 57, right? 57 years old. If the woman, if this woman is not this age, if this woman is copy and paste, if this woman is, that's darker, so this age, 40. So it's 40. So it's 40. Wow, that's long. Why is that? I have no idea. Why is that? Let me do it again. Maybe that's okay. So that is the risk. And if this woman is 40 years old, then it will have a chance of what? Say 100. How about that? Just 100. Make it easy. 100. Then which risk is higher? This age, right? This age is 40 years old. The risk to have a baby with trisomy 21 is 1 in 100, but the miscarriage due to the amniocentesis is 1 in 350, then it will be recommended. So typically that patient with 35 years old and older is suggested to have, because the risk to have the miscarriage by the amniocentesis in the new data is 1 in 1,600. So that is 1,600. So that is actually pretty low right here, right? Yeah. So that risk is always lower than the trisomy, the parent, the pregnant woman to have the trisomy 21. So any age is always safer to do a amniocentesis. All right, so that's that. So here's the summary about this important concept, including that karyotype is a technique to is a term, is a science to display chromosome in the order of the size. So from the 1 to 22 is by their size. And fetal karyotype can be used to detect the trisomy, especially the trisomy 21. Cell genetics analyze using staining and probe. So basically using a different type of staining to examine the chromosome. So it examines the chromosome compared to the karyotype. Here, it examines chromosome in more detailed, more molecular level. Chromosome shorthand can very quickly summarize the chromosome abnormality or chromosome situation. Ideogram display the feature of individual chromosome. So ideograms is this one. We show this one the very beginning, ideogram, just quickly here. Ideogram. So a graphic way to a schematic way to present the location of these bending and the genes, location of the genes. So that's not where were we? We are here. All right. All right. So so that's the karyotype in a way to analyze the chromosome abnormality, aneuploidy, basically. Now let's look into more of this chromosome abnormality, structure defect. Say this is a normal chromosome, right? And then we may have a deletion. We may have a duplication, like A, B, C, D, E, F, and D, E, F, extra. We may have the inverse. So where is that? The A, B, C, D, E, F, supposed to be D, E, F, but D, E, F inversed. So, and a typical one is the translocation. Translocation happens between non-homologous. Basically, that's because if it's homologous translocation, it won't make a difference, right? So say this part translocated with this part, then it will basically not going to appear as any abnormality at all, because you may thought that it's coming from this originally. But if this because the allele basically is the locus is basically not changed, it's the same, its alleles are different. But if this translocation happens to with say 1 with 3, then you will have the non autosomal non-homologous pair of the chromosome. And that could be an issue after the production of the gametes. We will look into that. All right, so this is the translocation that between the non-homologous pair, and there will be parts exchanged, and this translocation, if it breaks a gene, so this translocation, if it happens to be breaking the entire gene, then of course that gene will be affected. Or if it leads to a duplication and a deletion, we'll look into that. So that will cause an extra gene, right? And that can, all this will cause a condition called an unbalanced translocation. It could be a balanced translocation. So we basically switch one to the other with the other, but overall, the genes are preserved and everything is okay, even though their location are different. We will look into that. All right, we will have some figures showing you what's the balanced, what's the unbalanced. There are two types of translocation. One is called the Robertsonian translocation. The other one is the reciprocal translocation. This translocation lead to the balanced, unbalanced. Balance is the total genome, total number of the protein encoding gene remain in place, even though their locations may be different, but the total amount of the genes remain the same. So the cells can still express these proteins when it's needed, and the function of this organism is not affected. But it could be the deletion and duplication or gene breaks, then it will cause the unbalanced condition. So let's first look at the Robertsonian translocation. With this Robertsonian translocation, this happens with this acrocentric chromosome. So we talk about there are four major types of this chromosome. They are classified based on the short arm and the long arm. Now with this acrocentric, short arm is very short before the next step, which is totally disappeared, right? Or barely visible. And with this condition that's acrocentric, that short arm, the P is very short. And what happens with this one is that, say so two non-homologous pair have a breakage in the centromere. And when they are break, these two ends get sticky together. So this long arm put into the long arm of the other one. So these two long arms form a new chromosome. And what happens to these two short? These two short, they are too short, so they get lost. They got lost. So that's the translocation, Robertsonian translocation. So as you probably can think about this already, this will very quickly lead to monosomy, right? This will lead to the monosomy, a loss of the chromosome. It was before two, but now become only one. So you lost one chromosome. And so the patient will have 46 beginning, but lost one become 45. And this happens in only between these 5 acrocentric chromosomes, 13, 14, 15, 21, and 22. So that's that. This could lead to a balanced and non-balanced. So after this happening, after this happening, it will become 40, 45. The patient with the sometimes this short one doesn't really provide essential protein, so the patient probably won't show any phenotype. But the patients with the person with this Robertsonian translocation, it will carry this, and so in this carrier, there's no noticeable defects in the way that because this short arm doesn't really carry some essential genes. And then but the offspring may be affected, so the carrier may produce unbalanced gametes, and that will lead to the disorder in the offspring. It could be the abortion because they will not survive, or defects, and it survive but defects. So let's look at this balanced, unbalanced gametes. Say this is normal person. You have, say, chromosome 13. That's one with acrocentric. Chromosome 21. That's another one with uh, uh, acrocentric. You have the other one that is the long arm are attached. So this one, long arm, two long arm is attached, and this one is a 45 translocation, 43 translocation, 13 and 21. So that is that one, 31 and the 21. All right. So that's right here. So this one is the deletion 13 delete, and all right, so that this DER refer to the derivative chromosomes from translocation is present. Translocation of these two is presented. Q10, 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 Q10 refer to the remaining one. So that's derivative of the translocation of 13 and 21 of the result of this connection of Q location Q10 basically located those Q long arm and the long arm. So that's that. So that's this one here. So that's this one. This is chromosome 21, chromosome 30. Now with these two, the gametes could be right. Could be that. Could be. Let me draw here. Draw here. All right. So that could be this one, right? And this one with, so this one, right? And or lost one, or lost one, right? Right. So that's that one here, right? So it could be that this one here, and sorry, this one, right? So this would be this one, and this one, and this one, and this one, right? So that's that. Then you have these 21, 21, 21. You have 321. You have a condition like a trisomy 21 Down syndrome. So you have it's not exactly trisomy, but this long arm, basically you have 3 sets here. So that's that. So this is the typical example of the unbalanced gametes. If you have, for example, this is a balanced one, you may have this one, and this one, right? And this one. So result is balanced. So that's that. This one is also balanced, right? Because you have the like this one is balanced, right? You have 221 and 213. You have two long arm of the 21, even though you miss one short arm here, but that short arm really doesn't really provide essential proteins. So the person may appear okay, but as a carrier. So that is an example about that persons with a balanced, however, has the uh, uh, Robertsonian translocation provide the offsets with the basically a good chance to develop the Down syndrome. All right, thank you. So that's that. All right, so this is another example that's not 21 and 13, but 21 with 14. So as you can see here, we also have the chance to develop the trisomy 21 Down syndrome due to the unbalanced gametes of the Robertsonian carrier. So that's the Robertsonian carrier, Robertsonian translocation. Now the other one is called the reciprocal translocation. So with this one, it's not the break does not exist in the centromere, but the break exists in just in the middle of the long arm usually. So that's that. So for example, that these and these, they got translocated. CDE going here, MN going here. So it's reciprocal translocation. There was a difference between this one and the Robertsonian. Robertsonian lost the number of the chromosome. It will become the monosomy. With the reciprocal translocation, the amount of the chromosome remain the same. This can also lead to a balanced carrier, balanced that translocation, but balanced. It can also lead to a non-balanced partial trisomy of one, partial, not exact, partial monosomy of the other one, not exact. So it's partial monosomy. This one still provides some portion. So that's that. All right, so we basically complete all this chromosome abnormality. Now, the next step is to talk about DNA profiling. So we will finish this part to the end. All right, so that is something very relevant. People talk about it like very, very often. You see the news. But after the lecture, you should know what does it mean, the DNA profiling. What does it mean by using DNA to detect the suspects, to identify the suspects, and using the DNA to identify the remaining from the disaster. You have the disaster, right? And you have a piece of the body. How would you identify this piece of body belong to which person, right? You need to be able, you have the piece of body and you can use that DNA from the cells and you can conduct some type of the matching this DNA and that DNA. But what exactly do we match? All right. And are we all the same? What is a part that can tell us apart? So what's a part can tell us about in the genetic level? So that's, and what's the technology that we usually use, the typical used one to set us apart. So that's that one. All right, so here is the DNA profiling. We are looking to the length of the repeats. So this is a tandem repeats that that the repeat happens right next to the other one. So for example, GCATC, so this is typical repeats that we see it many times. And say, and the units, the sequence is very much the same across species. So this is very well preserved sequence. But the repeat number, the amount of these repeats vary between individuals. All right, so that is that one. And so for example, individual with, and each chromosome, each gene has two allele, one from mom, one from dad, right? So you can have the heterozygote repeats, like this one here. You have two repeats in one chromosome, the other repeat on the other chromosome on the other DNA. So if we have the individual one married with individual two, these two chromosomes happens to be identical in that repeat. This may not be the same in other repeats, but in this locus, that it happens to be the same. So this is homozygous. The let's see, what's it that individual one, individual two could have two possible, right? Could be 4 and 3 or 4 and 2. So if these if these two person has a kid, it will either have 3 and 2 or 3 and 3. So that chromosome pair or the repeats pair could be either this 2 and 3 or 4 and 3. If now you have a baby, this 4 and 1, 2, 3, 4, 5, 6. You can be pretty sure that this is not their baby, not their offspring. So that's a very obviously, very easily understandable way to tell if this infant belong to these couples, right? So that's that. Now, what's the technique we can use? This technique use first, we need to be able to, it's easy to see it in text, but the way that we need to do is to be able to cut it, right? We need to be able to cut it, and then we can weight it. Simple way to think about it: put it on weight and weight it. This one weight 2, this one weight 4, this one weight 3, right? And or the way we do it is using a gel electrophoresis. For example, we put this, we cut it, right? We cut it. We first need to identify this site. And we can do that. And we cut it. We can do that. And then we put this fragment in a gel. And we put 2 electrode here, a positive one side, negative on the other side, because these fragments are negatively charged. So they will get attracted into the positive side. So the smaller one will go fast. The heavier one will go slow. So we can see their profile. Someone will go further, someone will go slow, and then we can see if they are the same weight. If they are the same weight, they will go equal to the equal location. So that's that. So so let's look into the detail process. So here is kind of like graph showing you that we first got the sample cells, we got a DNA chromosome, then we cut it. It's not like randomly cut. We will selective cut on this side. So we will remove this fragment. and then we can go through this gel electrophores and to identify their location and to match if they are the same or not. So the way that we cut it is we use an enzyme. This is called a restriction enzyme. They are about 60. Sorry, why is this line here? There are 600 naturally appeared restriction enzyme. We can also create artificial one to match the one that we need. Then it will cut into the fragment. It will cut on this location and that location. Then the fragment is called restriction fragment length polymorphism, or short for reflips. And then we use this reflips run through the gel electrophoresis, and then we compare their location. And that indicate the weight, the molecular weight of each of these reflips. So that's that. Now, before we look into it, let's explain what does it mean. What's the meaning of the polymorphism in the genetic world? So this term means poly means multiple, morphism means form. So basically, this means that in the wild type, in the general population, we have different form in the gene, and that is basically that's what it means. So let's look into that. Polymorphism has two major types. One is called the single nucleotide polymorphism, or SNP. So that's that one. So basically, this gene and this gene and this gene are all the same. Only one part is not the same. So that's the single nucleotide. You may see this as a mutation, right? You may see this as a mutation. So what's the difference between SNPs and a mutation? Well, SNPs is a mutation. It's a variation. So DNA copy from one to the other. We earlier talked about there could be a genetic like error, like gene error, DNA errors, and we can fix it. The fix cannot be fixed back to the original form. So after the repair, we may form a new SNPs. Then what's the SNPs with mutation? SNP is a mutation. We don't call it a mutation because it doesn't really cause the disease. And because it doesn't really cause a disease, these gene survive. It will survive in our society, generation after generation. This one will have its offspring. Then we have a good population carrying these SNPs. If this cause disease, then this offspring will not survive. So if for those who barely survive, it will show the phenotype, and they probably won't have the offspring. So that's the mutation. And because these SNPs survive, they don't really cause disease. They may cause variation, but they don't cause disease. And then they will have a good population. This population we set a threshold, say 1%. If we have this polymorphism appear to be more than 1% of our population, demonstrate that they can survive, and so we call it a SNP. So we don't call it a mutation. So that's that. And another polymorphism is due to the tandem repeats. So for example, like this one here. So it's short tandem repeat polymorphism. So this is not right. And this polymorphism is the one that we talk about here. This is cutting of this polymorphism. So we cut here, and we cut if we can cut here. All right, so that's what we do. We cut this and cause a fragment called the reflip, and run using this fragment to run through the gel electrophoresis and compare their weight. So another, and so say this is result, right? Say this, if we look into that, this is a famous one that we want to demonstrate that the clone of a sheep that was about 10-20 years ago, that first cloned multicellular organism from a sheep. This sheep is called Dolly. So we get the DNA fragment and we run through this gel. So let's look at that. 1, 2 to 12, that is 12 other ships. D is the produced Dolly, that is the cloned ship. U and C are from the U is from the donor, so donor donate is a cell to produce the Dolly, and also donor cells get cultured, so we have DUC. If Dolly is the clone of this donor, then we expect to see their DNA profile to be identical, and that is what we saw here. So here is the DUC. U is the donor. C is the culture cells from that donor. D is the Dolly. So you can see that they basically identical to each other. So their reflips have exactly identical molecular weight. So that's that. But very different from other ships. So let's look into the procedure here. Basically, we will talk about the major thing is the cutting restriction. Why do we restriction enzyme? So this one is the key. Let me bold it. This one is the key, restriction enzyme. Without it, we cannot do it. We cannot have a precise cutting. If we have it, then this technology become doable. So do we have it? Yes. Where do we find it? Originally, this enzyme, this restriction enzyme was originally found in the bacteria. So the reason is that the bacteria will use this to cut the DNA of a virus. And what they do is that they first kill that virus. Then the second is that they will preserve this segment and memorize it so the cells will recognize this virus in the future. So that's their protective mechanism. And we later observe this enzyme, then we can know that this is a good way to locate a specific site in the DNA. And in addition to identify that site, we can cut it. So that is a very good tool that we can use. So we have that. So this enzyme is called restriction enzyme. Is because that it restricts the virus intrusion. So it restrict that. That's why it's called restriction enzyme. Further, we also call it endonuclease. So basically, that is a breakdown of the DNA nucleus, endonuclease. Originally found in the bacteria, and they digest the virus. So that's for example, this is the E. coli restriction enzyme. They cut it, they cleavage it, so they can break it. Specific site, specific recognition sequence. They recognize it and cut it. So it can cut it at, it's not exactly at, That's why a newer technology was also developed, like the one CRISPR Cas9. That's the one provide more accurate cutting. This cutting, even though that we say it's very precise, in the reality that it can recognize it, but when it cut it, it's very brutal. It's not that precisely cut. If we say that CRISPR Cas9 is the one, the newer one is the cutting that you and me cut a piece of paper. This restriction enzyme cut it like 3 years old cutting a piece of paper. They can cut it, they can recognize the line, but when they cut it, it will be pretty brutal. Okay, so it's not very precise, but good enough. If we wait, it's still good enough. So that's that. All right. So this site, the recognized site, sometimes it could be a palindromic sequence. So like this one here, like this one here. The palindromic sequence means that when we read it back or forward, they will read the same. GCA, TT. Sorry, that probably not the good one. So let's look at this one. GC. Sorry, that's right. So that's a good one. GAATTC, GAATTC. So that is, we read forward, backward. That's the same. That's the same one. So that's this palindromic sequence. There are about 3,000 restriction gene enzymes available from the bacteria. More than 600 is already available in the commercial. They are good at cutting the DNA, doing the DNA modification. So we have this enzyme. And what we will do next is to cut it. This enzyme cut DNA into two types. One is called a blunt end. It's called protruding end. So the other one is a staggered cuts, produce a sticky end, cohesive end. So let's look at this one here, easier to understand. So this one, two type of DNA cutting from this restriction enzyme, we can cut it, leaving some sticky end. So it can be attached to the other things, so it can be sticky end, or it could be the blunt end. So that's that. In addition to this naturally exist this enzyme, we can also design the one we want. So this is called the artificial restriction enzyme. So basically that we can have the desired sequence to be cut, and we can use the restriction enzyme using part of it and put these two together so we can identify the site and using this restriction enzyme to cut it. This is a typical one. The most common one is called the zinc finger nucleus. And so this one is, for example, a figure is like this one. The zinc finger means that it uses zinc in this molecule, and these fingers, I remember like 7 fingers. Wow, more than that. All right, several fingers. So it will basically fingers will have each one attached to a particular sequence and then pull it open, and then they can cut it. So that finger is a good way to identify the gene and then separate it. So that's that. And usually that will come with the combination of the binding domain with the nucleus. We typically in this zinc finger nucleus, they use this type 2 restriction enzyme called focal, FOKL. To There are about 4 types of the restriction enzyme. That one binds with this DNA binding domain and can be used to cut the site that we desire to. So that's that. All right. So that basically is a technique that we can use to cut these different tandem repeats. And then we can weight it. We can measure the length and to distinguish if this DNA match with the other DNA. And we can use that DNA to identify if these samples match with the other person's cells. So that's that. And of course, this techniques has been become very useful in terms of the forensics, right? Identify the remaining piece of the body with the one that you want to match. We can say, for example, after a disaster, that we want to locate whether we want to know that whether this piece of body belong to one of your relatives, then you can get the sample, even though you don't have that relatives here, but you can get there like a toothbrush, then we can see if there's remaining tissues there. Or the worst scenario is basically just check with their blood relatives, blood related relatives. And so we can have 2 samples and compare their compare their what's that? Oh, I want to get the term right. Oh. Compare their so this technique can be used to identify to conduct the DNA profile. So of course that this has been used in very commonly. So the typical protocol to conduct DNA profile is to use these 13 short tandem repeats. So here showing you, I think we showed these slides before, right? So very typical one that 13, the screen identify this, the location of these repeats. Everybody has this 13, everybody has the same sequence, it's just the length is different. And this length different is called the variable number tandem repeats right here, variable number tandem repeats right here. So I bold it here. And so we use that. You may be lucky that this repeat 2 people have the same number, but if you want to have all the 13 number to be identical between these 2, the chance is 1 in 250 trillion. So it's almost impossible that 2 people are identical with all these 13 repeats. And the way that we can quantify the chance of that, right? So so the probability. So basically, we can compare this one with compare 2 person to see how possible these 2 is the same. Say if we only have one repeat, and if this repeats to be only if this repeats to be different, then here is a frequency. How do we know this frequency? Basically, we know this frequency based on human genome, gene-wide study, right? So we have the population. So we know that the population with these repeat located in the chromosome 4 is this frequency, that only 1 in 60 have that number. And so that's a frequency. And if you want to have that identical, and if you have the second one identical, this one say frequency 24, 1 in 24. So you have the frequency for each individual based on this gene-wide study. And the way to do it is you time it up. So if you have all these 6 identical, the chance that this is not. So how possible you two are the same if you two are not is 1 in this, right? So 1 in overall, if 13 is 1 in 250 trillion, meaning that only if there are 250 trillion people in the world, then it's possible that there are 2 different people are identical, right? Meaning that it's very, very unlikely. So that's the frequency. And in the end of these 2 slides, I want to show you how people quantify this frequency. So very quickly, that we need to know the equation. This equation is called the Hardy-Weinberg equation. So this equation is very interesting, that this is a paper published in Science. You probably didn't know it, but the journal Science is actually a pretty good journal. And this is a mathematics is something like a mathematic profession. They try to explain a condition in the genetics. And so they come up with this equation, very simply understand. So here we have, say, two group of allele, 2 type of allele, P and Q. And say this is one allele, this is the other or others, say P and Q. You don't need to worry about the recession possession dominant, recessive dominant here. So basically it's 2, right? Say we have only 2. You can be this and the other else. So it could be overall that these 2 binds together is 1. So it make it a whole. The chance is 100% if these 2 apply. P is this portion, Q is the other portion. So the overall is 2. And a simple math, we can know that with this equal to 1, this equation will also hold true. P squared plus 2PQ plus Q squared equals 1. And that's also holds true. So basically that, say you have PP means P squared, QQ means Q squared, PQ, so you have P squared plus 2PQ plus Q squared equals 1. So that's that. And so and you with this in mind, we can quantify the percentage. So first of all, you got a sample. And you got a sample, you must know that each short tandem repeats short tandem repeats STR has 2 allele. These 2 allele could be homologous, homozygous, heterozygous, or it could be the homozygous, right? So for example, this allele, this is heterozygous. So you can have one is, we know that frequency is 0.6, the other frequency is 0.3. And so the overall frequency is P and Q. So you got 2PQ. This frequency basically is this one, P and Q. So you have 2. So the chance to have P and Q is pretty high, right? It's almost half. PP, QQ, if the person with PQ the chance of the offspring to have PQ is half. So you PQ times 2 plus times 2. So that's this one here, 2PQ. So you have 2PQ. If it's PQ, you got 2PQ. So the chance of this one is 0.3, 2PQ, 3 times 6 times 2, 0.3 times 0.5 times 2, and PQ 0.1815 times 0.8 equal times 2, 0.024. Now you have this one. This is homozygous. And the homozygous, you need to do this one square. So 0.2 square equal this one here. Again, heterozygous. So you have 2PQ. That's this one. And overall, you times them together, and then you got a chance of this 5 sequence. So that's that. And if we have chance, we probably can do some practice on this one. If not, this is the example that I provide you. So that ends it. And this is the chromosome variation. And this is how can we use this chromosome variation to conduct a DNA profile. All right, let me know if you have any questions. And that ends it. Let me see if I can do something about it. Please. Stop sharing first. Don't die on me.